All right, so we can go ahead and get started. Hello, everyone. My name is Miss Jessica Finn.
I'm Ms. Kelly Van Horn.
And we would like to thank you so much for coming to our first ever virtual science research symposium. We hope that you and your families are doing well during this very crazy time, and we really do appreciate you taking the time out of your day to come and join us and help us to celebrate our seniors and showcase all of the work that they've done in the science research program. The seniors who are in the program, they've been working really hard on their research for the past 3 years that they've been in the program. And even though we can't come together as one whole group and have our traditional kind of symposium, we are really, really ecstatic that we are able to celebrate them and what they've been able to accomplish in this kind of virtual setting tonight. So again, I thank you so much, and we hope that you enjoy.
So as Ms. Finn said, this is our first ever virtual symposium, and we really do appreciate everyone coming out to see all the work our students have done given the current circumstances. This evening, the juniors in our program will introduce the senior presenters. While the students are presenting, you can type any questions you may have into the Q&A, and the students will answer all questions at the end of their presentations. So to start us off this evening, here is Natalie to introduce our first senior presenter.
Hi, first up we have Francisco Vazquez, and he will be presenting on EEG data analysis for stroke patients using VR rehabilitation, and he will be going to NJIT this fall.
Hear you.
Can you hear me now?
Yes, perfect.
Okay.
Go get him.
All right. Thank you, Natalie. Hello, everyone. So, there you go. So I'm going to be presenting on EEG data analysis. This is research I did over the summer at NJIT. Okay. So stroke. So stroke is a blockage in the brain that causes blood flow. So it's when part of the brain isn't getting enough blood, which means it's not getting enough oxygen. So therefore, those cells in the brain start to die off. Usually it occurs on one part of the hemisphere, so one side of the brain. So stroke rehab is something that is evolving. So it involves improving muscle strength. So the brain controls the whole body and controls muscle activity. And when part of the brain dies and is damaged, you lose muscle strength, you lose coordination. So stroke rehab consists of regaining that muscle, regaining that coordination and that range of motion. So EEG, so what is it? So it's a cap and it records electrical brainwave patterns using electrodes. EEG, its main purpose is to check for desynchronization. So in the middle picture, this is what a stroke is. So it's usually one part of the brain is blocked off and it damaged, and since the other parts of the brain aren't getting oxygen, they die off. On the right, we have a mat that is used to help patients regain muscle strength in their legs to help them improve walking again. And on the left, we have the EEG, the cap. So each one of these are electrodes, and they are used to check for desynchronization. So these two graphs in the middle, this is what you see on the computer. So each one has numbers on the sides, and these are representative to electrodes. So each number is an electrode, and these waves is what they're reading. So they read the electricity in the brain. On the left side, we have a clear view. So the electrodes have 100% connectivity, and on the right, it looks more fuzzy because there's some type of disturbance. So on the left is basically what we want to see. That's good data. So we checked for the beta band, which is a location in the brain, and it's found over the motor cortex, and it has frequencies of 12 to 30 hertz. So that's what we focus on, that frequency. So as I said, EEG checks for desynchronization. So what is desynchronization? It's suppression of waves. So in the brain, when you're at rest, there should really be no movement in the brain. It should be calm and relaxed. It's when you trigger a muscle from blinking your eyes, moving your eyes, flexing a muscle, your brain sees spikes, and that's a desynchronization. So that's what the EEG checks when you activate something in the body. So my hypothesis consisted of the dominant hand will have more desynchronization than the non-dominant hand. So if you're right hand dominant, your left hemisphere, so again, so actually in the brain, the opposite hemisphere controls the opposite side. So the left hemisphere controls the right side of the body, and the right hemisphere controls the left side of the body. So in my hypothesis, if you're right-handed, your left hemisphere should desynchronize more. So that's basically what it's saying. So the materials that we used an EEG with 64 electrodes. There are different types of EEGs. So there's ones with 32, 64, and 128. We used one with 64. So we also used EEG Lab. So that was a software on MATLAB on the computer. We also used Oculus Rift, which is the VR headset that we used. So now the methodology. So I'm going to show a quick video of what the actual experiment looked like. So as you see, he's the one that made the program that you see on the computer. So currently he's wearing the EEG on top of his head, and he's wearing the VR headset that's connected to the computer. So on the screen, you see his virtual hands and you see the red lines. So on the screen, it prompts him to move left or move right. It says prepare, and then the red lines move, and he has to move his hands to go on top of the red line. So that's basically what the experiment consisted of. And the EEG is recording the desynchronization that occurs in the brain while he is performing the tasks that are shown on the screen. So the methodology. So the electrodes are placed in salt water for 15 minutes. So there's two types of EEGs. There are gel-based, and then there are plastic-based. Usually people don't like to get gel in their head, so we decided to use the plastics ones, and we have to put them in salt water for 15 minutes so that they're more receptive to the brain patterns in the brain. So we place the EEG, so we have to measure in between the nasal and the inion. So basically we have to measure on top of the nose, so in the front, and on the back of the nose. So as I was saying, so you have to measure on top, basically right above the nose and right below the nose, and you want to make sure that the center is right in the middle. So basically where CZ is. And this graph or this picture is showing the impedance value. So before you start collecting data, you want to make sure each electrode is green, that they're all connected and that they're all getting correct data. If any of them are red, that either means that there's too much hair, so you have to spread out the hair, or that it didn't have enough salt water, and basically you just have to add more salt water so that it's more receptive to reading those brain waves. So we clean the hair and remove dead skin using the alcohol wipes. We place the center. We make sure all electrons are connected using the impedance value. So we want to make sure it's green. So we also have to test that we're getting the reading. So we ask the patient to blink and see if all the channels are working correctly. As I showed in the graph, we want to make sure all the waves are correct, that there's no noise. So then, in order to make sure there's no noise, we ask the patient to bite down, and we should see a spike on the waves on the graphs, and we want to see that. So we also need a ground reference. So in the video, he was wearing a wristband, and that was basically the ground control. So in this experiment, we used C3 and C4, which were they're basically in the middle of the line. So these two, C4 and C3, were the two electrodes that we used. So all the other ones were basically just to check for noise and to check that the readings were correct. But the ones we were focused on are those two in the middle. So we had 200 trials. So in the video, you could see on the screen that it was counting down. So we did 4 sets of 50 for 1 minute rest. So we kept the headset on throughout the whole experiment. So throughout the whole experiment, we were collecting data, and we only did 200 different trials. So 4 sets of 50. On the screen, it said prepare to move right or left, and then the red line would move, and then the patient would move his hands to go onto the red line. And this is exactly when synchronization was triggered. So we calculated epoch of negative 1.5 and 1.5. So this is basically a second and a half before the command happened and a second and a half after the signal occurred. That's the data that we specifically wanted to see, because when you see the cue on the screen, your brain gets triggered. And then once you move your hand, that's when actual desynchronization occurs. And that's what we wanted to see. So after we collected all the data from the 200 trials, then we have to filter out the data, because the patient wasn't completely still throughout the whole data. So there were noise in some of the data because they have a tendency to flex a muscle, either move their fingers, move their feet, look around with their eyes. So that produces noise, and we don't want that. So we want readings where it's purely brain activity. So we ended up removing 13 trials of the 200 trials. Then we filtered the data from 1 to 50 hertz. So as I said in the beginning, the beta band is from 12 to 30, but first we had to filter it to a reasonable size of 1 through 50. So we kept all the brain activity and removed all the data that had noise. So we had to reprocess 1 through 50. So the average of every time point frequency uses a beta band 12 through 30, as I said. So that's the frequency ratio. So then after we remove the trials that have noise, we check for desynchronization. So on the computer, we have to go into EEG lab, and we see these maps. So this is an example of one where we have noise. So on the right side, you have different categories. You have brain activity, muscle activity, eyes, heart, and nose. So on the top one, you can see that's basically they were moving their eyes left to right. That's what the data is showing. On the second one down below, where you see the red heat map, they were blinking their eyes. And on the left, what we want to see is red and blue spots with no green horizontal lines. So as you can see, they all have these greenish yellow horizontal lines, and that's noise. That's something we do not want to see. This next one, on top we have heart rate. So it was picking up its heart rate. That's why you see red dots, because it's the pulse basically. The one down below was muscle activity. So it was most likely he was either biting down or just moving his fingers or hands. These next two are perfect brain activity. So this is exactly what we want to see. So as you can see on the right, it says brain 99.6 and brain 81.5. So this is exactly what we want to see. These red and blue dots that happen consistently throughout, and we want to see the spread of the color from red to blue. That's exactly what we want to see. Okay, so now the results. So the hypothesis was correct. The dominant hand did desynchronize more than the non-dominant hand, and the opposite hemisphere to the hand that did desynchronize, it did desynchronize more than the other hemisphere. So as we can see, so these were left-hand trials. So these were the trials where the person moved their left hand. So on the left side, we have the left hemisphere, and on the right side, we have the right hemisphere. So as I said before, opposite hemisphere contracts to the opposite side of the body. So on the left-hand trial, my hypothesis said that the right hemisphere, which is on the right side, would desynchronize more. And we could see that the blue pattern is a lot stronger, a lot more vibrant, a lot bigger. So that means that it did desynchronize more on the right side. And again, this is the right-hand trial. So my hypothesis said that the left hemisphere would desynchronize more, And again, we can see that in the size of the blue pattern. So these are the graphs that show the same exact data as we saw before, just in a different format. So these were the left-hand trials. So my hypothesis said that the right hemisphere would desynchronize more. So as we can see, where we want to see the desynchronization happen is between 375 and 400. So that's where we see the red line, the right hemisphere. It desynchronizes a little bit more than the other side, which is very good. And again, with the right-hand trials, the left hemisphere should desynchronize more. So this data, you can see it wasn't the best. So the blue should have gone down a little bit more and earlier. So there was some bad readings that caused for the right-hand trials not to pick up the data correctly. So that most likely means that the electrodes that are on the left side of the brain weren't connected as they should have been. So the discussion. So some of the cons were that there was a lot of noise in the atmosphere. So we did this experiment in a lab with a bunch of different people. So there was noise inside the atmosphere. What we could have done was had the patient be in a room by himself. Therefore, there would have been a lot less noise. And again, the left hemisphere dropped in the left-hand trial. So that could have been because the electrodes weren't 100% connected. Either they could have ran out of salt, so we could have added a little bit more salt water. So pros to the experiment. So the reason why we wanted to do VR rehabilitation is because it can be done at home. So someone can purchase a cheap VR headset, can download the app, and they can be doing this rehab at their house. So they don't have to go to a facility. They don't have to pay a lot of money to get this rehab. So those are the pros to this experiment, this method of rehabilitation. Some future studies are virtual reality versus non-virtual reality. So there are headsets where you don't see virtual hands, where you actually see your real hands. So that could be tested. And another one is the mirror test. So as I said, desynchronization is when there's suppression to the wave, so you're activating muscles, but it also occurs when you see something. So when you see someone move their right hand, inside of their brain, their left hemisphere is being activated. But in your brain, your left hemisphere is also being activated just by looking at the movement. So another future test is basically putting a mirror horizontal to the patient, where they move their right hand or their left hand, and they see the mirror where basically they're projecting that their left hand is moving, even though it's just a reflection of their right. So that would trigger the opposite hemisphere in the brain. So that's another test that they wanted to work on. So in conclusion, stroke patients have less desynchronization. So we did this experiment with healthy people and with people with that had previously had a stroke, and the desynchronization did occur more with healthy people. Another conclusion is that one hemisphere will always be weaker than the other. So again, the reason why we have a dominant hand is because one hemisphere is a little bit weaker than the other. And in the end, this rehab will improve the affected hemisphere by just repeating the trials and repeating this rehab program. So I would like to thank my mentor, Dr. Adamovich. I'd like to thank my teachers, Ms. Finn, Ms. Van Horn, and Dr. Brickman, for giving me the opportunity to really learn and research the things I enjoy. So now, if there are any questions. Did you see any improvement in desynchronization after the many trials? Was there evidence of rehabilitation using this VR program? Also, how can you reduce the atmospheric noise? So the best readings were done at the beginning, because that's when we had just removed it from the salt water. So as time went on, the readings got, they were basically getting less and less correct readings because we didn't use gels and we used salt water. Was there evidence of rehabilitation using this VR? Yes, there was. So I didn't include that in this presentation, where we focused on the trial specifically towards people that had strokes. But yes, this program, and there are other games and programs using VR. So there's one called the block test, where basically you have a VR, a box that's split in half with basically a bunch of building blocks, and you use them to see how many you can pick up within a certain time limit. So 30 seconds or 60 minutes, and see how many blocks you can move from one side that's full, one side that's empty. So that's another game. And yes, this does really work. And how can you reduce the atmospheric noise? So we were in a room with multiple people and multiple computers, so that drastically increases the noise. So if we were in a room with just that one computer that's doing the reading and just one person, that would definitely drastically reduce the noise. How does this present of hair disrupt the EEG data? So as I said before, you want to make sure that the electrode is firmly placed on the skull, and hair basically provides a barrier, and it makes it farther for the electrode to be from the actual brain waves. So it creates a disturbance. So basically you just move the hair and make sure that it's on the actual scalp. So if there are no other questions, next up we have Anisha introducing the next presenter.
Montville Township Public Schools. Educate, inspire,